At the southern end of Africa's Great Rift Valley lies Gorongosa National Park, a vast, unexplored wilderness, devastated by civil conflict. Now, the park is joining forces with biologist Edward Wilson to create a research facility like no other, the E.O. Wilson Biodiversity Laboratory. Together, they're bringing some of the brightest minds in science to Mozambique, discovering Gorongosa's secrets, nurturing the next generation of champions, and restoring this once mighty ecosystem.
They say you never really know someone until you've walked a mile in their shoes. If that's true, my colleague Tyler Kartzinel and I are about to get to know ecologist Kenneth Tinley very well. Tinley came to Gorongosa in 1968 with the ambitious goal of mapping the ecological boundaries of the park. Over the next 5 years, he would observe and record every aspect of the natural environment and create a framework for the entire ecosystem. From the course of the rivers to the seasonal migration of the animals, almost all the scientific knowledge we have about Gorongosa from before the war came from his early efforts.
Now it's our turn. We're here to repeat some of Tinley's early vegetation surveys with an eye towards understanding how vegetation shifts may play a role in the low herbivore numbers in the park.
I think that the critical thing to understand is that the plants out here are the foundation of the ecosystem, and without having baseline knowledge of the plant communities, there's no way to contextualize the studies of the lions and the elephants and the zebra, and no way to anticipate what's going to happen to the ecosystem in the future.
It's really important that we understand what's happening with the floodplain. This is kind of the epicenter for all the herbivores here. And as the park plans to bring a lot of these animals back, it's important to understand whether there's enough resources for those animals to make that a sustainable solution moving into the future.
40 years ago, Ken Tinley did an intensive study in the Lake Urima floodplains where he documented the plant communities and the herbivores and the predators that live out there. And he provided a framework that paints a scientific picture of the ecosystem.
One of the other resources we have is old photographs like this. These pictures show thousands of zebras and wildebeest and elephants out here. And as we look out over the floodplain today, we see mostly waterbuck and warthog. And one of the main motivations for being out here is to understand how the extinctions over the last 40 years have changed this ecosystem. Using a GPS and hand-drawn maps, we can locate the precise locations Tinley surveyed all those years ago. By replicating his methods and comparing his findings with ours, we can start to get a sense for how the floodplain has changed over time. We've still only looked at a few quadrats, but as you can see here, this one's very different than what Tinley would have described. Tinley tells us that the dominant species in his plots were grasses like cynodon, but in this plot you can see it's dominated almost entirely by forbs, and the forb cover is really high here. And in the midst of all these forbs, you can see small grasses popping up, but this is definitely not what we would characterize as a cynodon lawn.
A second goal of our study is to work out, in the absence of abundant grasslands, what the dominant herbivores are now eating.
So we're hanging out here with a herd of waterbuck, and one thing that we really would like to learn is what the waterbuck are eating. And one of the most direct ways to figure that out is to look what comes out. So let's go do that. The next part of the whole process is to actually get the DNA out of the samples. I bring a lot of different equipment and chemicals with me and break the DNA away from the sample and separate it out. And at the end of the day, I'll have very pure DNA, which we can use to identify plants from the sample.
We've come out the other side muddy and wet, but with immense respect for the work Tinley did, and perhaps more importantly, what it means for the future of Gorongosa.
We know it's changed radically in the past, and we know it's going to continue changing. And anything that we can do as scientists to help understand the causes and consequences of those changes, develop a new framework for understanding how it's going to continue to change, I think will be extremely valuable going forward.
Small steps on a journey of 1,000 miles. Each new discovery bringing Gorongosa closer to its original vibrancy and hope for all of the world's wild spaces along with it.